Hello everyone, welcome to chapter 11. So now we're going to be diving into the nervous system and nervous tissue. So let's start talking about what nervous system is in the nervous tissue. So the nervous system is a master controlling and communicating unit or system of the body. Cells communicate via electrical and chemical signals, as you probably already know, and they're rapid and specific. And so basically they cause almost immediate responses. And of course, the delay between that communication, that electrical and chemical signal, even there's a delay, is nanoseconds. It's almost immediate response. So we a lot of times don't know the difference between that signal being sent and being produced. So the nervous system has three overlapping functions. It has that sensory input, so that information that's gathered by those sensory receptors, sort of about the internal and external changes. And then the integration, and that's the process and interpretation of that sensory input. And then there's that motor output, so the activation of effector organs, muscles, glands, things like that produces that response. And so essentially here in this image, you can kind of see the nervous system functions. You see a glass of water, you get a sensory input, that it gets integrated into the nervous system. You're thirsty, you want it, and that motor output is to reach and grab it. So basically, the nervous system is divided into two principal parts, that central nervous system and the peripheral nervous system. So the central nervous system, or the CNS, that's the brain, the spinal cord of that dorsal body cavity. It's the integration and control center. So it interprets sensory input and dictates motor output. And of course, the peripheral nervous system, or the PNS, is a portion of the nervous system outside the central nervous system. So that contains the spinal nerves to and from the spinal cord, and the cranial nerves to and from the brain. So you can see the difference here. You have that central nervous system, that brain and that spinal cord, and you have the peripheral nervous system, which basically consists of those cranial nerves, spinal nerves, the ganglia, and so on. So the peripheral nervous system, or PNS, has two functions. It has this sensory afferent division, so somatic sensory fibers. That's where you have that convey those pulses from the skin, the skeletal muscles, and joints to the central nervous system. And then you have visceral sensory fibers, which convey impulses from the visceral organs to the central nervous system. So that's that. So those are those two aspects of that afferent or sensory division. Then you have the efferent, or the motor division. And the motor division is responsible for transmitting impulses from the CNS to the effector organs. So that's things like the muscles and glands, and of course the two divisions, that somatic nervous system and that autonomic nervous system. So the somatic nervous system is a somatic motor nerve fibers conduct impulses from the CNS to skeletal muscle, and a voluntary nervous system. It's conscious control of these skeletal muscles. The autonomic nervous system consists of visceral motor nerve fibers. It regulates smooth muscle, cardiac muscle, and glands. It's the involuntary nervous system. So it really has two functional subdivisions, the sympathetic and the parasympathetic. And really they work in opposition of one another. So this is sort of the overall organizational scheme or chart of our nervous system. So of course you have this central nervous system, or CNS, that's the brain and spinal cord. It's integrative and control centers. And of course, back and forth, that works with that peripheral nervous system, the cranial nerves and the spinal nerves, communication lines between the CNS and the rest of the body. Now, you can have this sensory afferent division, that's that sensory nerve fibers, that can work in the direction to the peripheral nervous system back to the central nervous system. The peripheral nervous system will also act on the motor, or the afferent division, which then will activate or cause a reaction in the somatic nervous system and autonomic nervous system. In the autonomic nervous system, of course, that can trigger the sympathetic division and the parasympathetic division. So you can see this organizational chart and how that sort of works within the entire body system. So we also have neuroglia. So nervous tissue histology is also very important. A lot of times we don't really think too much about the nervous system histology, but it's really important. There's lots of cells and tissues that make up that nerve. So nervous tissue consists of two principal cell types. The neuroglial, that's what we're called a glial cell. Those are the small cells that surround and wrap delicate neurons. And then we have the neurons, and those are literally the nerve cells that you're probably the most familiar with. Those are the excitable cells, and they transmit those electrical signals and things like that. And so basically those are those two subtypes. Now there are four main neuroglia that support the central nervous system: astrocytes, microglial cells, epithelial cells, and oligodendrocytes. And those are some of those cells that help support those neurons, of course. And so what you can see here is a neuron. And here is one of those astrocytes. And you can see how those astrocytes, they're the most abundant CNS neuroglia. You can see the wrappings around those capillaries and how they're really connected. So that's that support cell for that neuron. And also microglial cells, those are the defensive cells in the CNS. So you can kind of see, here's that neuron and here's that innervation of that microglial cell. We also have the epithelial cells that line the cerebrospinal fluid in those CNS cavities. So you can kind of see this is the brain or spinal cord tissue. And those the epithelial cells here are cilia, and there's a fluid-filled cavity. So you can see how that it basically unites or makes that connection to that brain or spinal tissue, to that fluid cavity. Neuroglia as well, so you can see things like the oligodendrocytes. Sometimes I refer to them, and I might slip back and it's oligodendroglial cells, but the oligodendrocytes have processes that form myelin sheaths around nerve fibers. So this is an axon, and here's that oligodendroglial cell that is forming this myelin sheath around that nerve cell. That's a sort of a protective mechanism, and those are really interesting, and we'll talk a little bit more about those. So you can see here, this is that body, the cell body of the neuron. You have some satellite cells that surround it. A myelin sheath, here we call this a Schwann cell. And so you can see that myelin sheath is formed around it. So satellite and Schwann cells, they form those myelin sheaths around those neurons in the peripheral nervous system. And they're very, very important in the protection of that nerve cell. So the neurons themselves, again, the nerve cells, they are the structural units of the nervous system. They're large, highly specialized cells that conduct impulses. They do have some special characteristics as well. Their extreme longevity. It lasts a person's lifetime. That's actually good, and it can be a detrimental thing as well. And by detrimental, I mean it's good that it lasts a lifetime. But it's because they last a lifetime, they're not necessarily regenerated. So once we lose them, they're gone. And that can be the troubling part of that. They're amniotic, with few exceptions, and they have a high metabolic rate, which requires this continuous supply of oxygen and glucose. And in fact, the majority of the oxygen and glucose that you have in your body basically is utilized by the brain and the nervous system. All have a cell body and at least one or more processes. So this is that structure of that motor neuron. So you have the cell body, of course, lots of little dendrites. Those are the receptive regions. The nucleus, of course, is found in the cell body. And then, of course, all those little parts of the cell we talked about, like mitochondria, endoplasmic reticulum, Golgi body apparatus, all found in that cell body. And then you have an axon, and axons are covered in these little Schwann cells that make that myelin sheath. The axon, it's the secretory region. It's where the terminals attach to those different bodies where the signal's being transmitted, the muscle, different things like that. So this is just the structure of that motor neuron. Here's that dendritic spine here, and then of course the neuron of the cell body. Now, PNS, the peripheral nerve system nerve fiber myelination. You can see what that myelin sheath looks like as it surrounds that axon under the electron micrograph, those Schwann cells that I mentioned. Myelination in the peripheral nervous system. These myelin sheath gaps, those exist between the Schwann cells, and let me go back and see. So you see that there's some gaps in between these Schwann cells. And so basically that is sites where little axon collaterals can emerge. We call these the nodes of Ranvier. And basically they're just little protrusions that can come out if needed from that axon. And then, of course, you have some non-myelinated fibers. Those are thin fibers that are not wrapped in myelin sheaths, surrounded by Schwann cells, but then have the little coiling. So one cell may surround maybe 15 or so different fibers. So again, you saw this just a minute ago, but that structure of that Schwann cell. The myelin sheaths in the central nervous system are formed by the processes of the dendroglia, not the whole cells. And each cell can wrap up to 60 axons at once. The myelin sheath I talked about is present, but there's no outer collar of the paranuclear cytoplasm. Thinness fibers are unmyelinated, but they are typically covered by little extensions of extensions of adjacent neuroglia. So the myelin sheath in the central nervous system are white matter. There's some of those regions in the brain and spinal cord with dense collection of myelinated fibers. And then there's gray matter, which is just mostly neuron cells. So the white matter is because they're covered in these myelinated fibers. So these are some of the classes of these, the structural classes of neurons, I should say. We have these multipolar, which basically says has many processes extending out of the cell body that you see here. All are dendrites except for this one single axon that's going to go on to where it's going to be connected to. We also have bipolar, and that is where the two process extends from the cell body. One is a fused dendrite and one is an axon. So you can kind of see that structure. And then we have unipolar. These are quite interesting because you have this little cell body, but basically you have an axon extending with the central process and peripheral process there. And so those are pretty interesting because they have receptive endings on both ends. So you can see what they look like. This is the receptive region, which receives the stimulus in purple. And then the trigger zone or conducting region here, basically in green. And then, of course, the red, which is the secretory region, where the stimulus is released. So you can see how different that is in each of these different structure types. So you have a huge trigger zone with regards to the bipolar neuron, where a very small trigger zone for a unipolar. And so it's very interesting to see the difference between these and how they behave. Also, when we compare these guys, basically we have how the structural classes of neurons work. The multipolar, you can see some have all these dendrites that branch significantly. And of course, this is found in the cerebellum. It's called a Purkinje cell. So they're most abundant in the body. And then you have these pyramidal cells. Basically, you see the dendrites look very, very different. You also, in the bipolar cell, an olfactory cell is one of those things where we can find in the eyes, the ears, the olfactory mucosa. You see the dendrites basically here with the cell body really close and long axons. Retinal cells, you have this cell body with a pretty long dendrite with branches and an axon. And then, of course, this unipolar that we talked about, the cell body's here and basically receptive endings throughout the whole process. This you would find as a dorsal root ganglion cell. Some more structural classes and how to look at this multipolar. Most multipolar neurons are interneurons that conduct impulses with the central nervous system. Here you see that with a muscle. You also saw this image in the last chapter when we were memorizing and talking about muscle cells and things like that. This bipolar type of neuron you can see in the retina of the eye and the impulse to the brain. And then this unipolar, of course, you can see that sensory neuron and that impulse for the skin. So it's really important to have these different classes of neurons that really help make up who we are, essentially, with regards to our responses to the environment. Now, one part about the nervous system that's very important is in the nervous tissue is the synapse. So the nervous system works because information can flow from neuron to neuron in this cascading-like symptoms. So neurons are functionally connected by synapses, and these are basically junctions that mediate an information transfer. So from one neuron to another neuron, or it could be a neuron to an effector cell. And so that synapse is really, really important. So the presynaptic neuron is that neuron conducting impulses towards the synapse. That means it's sending that information. And then the postsynaptic neuron is the neuron transmitting electrical signal away from the synapse, meaning receiving the information. So in the PNS, that may be a neuron, a muscle cell, or a gland cell, but most actually function as both. So you can kind of see here, here is the axiosomatic synapse here. So you can see these myelinated things. You've got the cell body here. And then, of course, you have some axiodendritic synapses, and you have axional synapses as well. So basically some different types of synapses that can occur within that system. Of course, this is just another image from a micrograph that's been colored to show you the different synapses. This, of course, is the cell body, the soma of that postsynaptic neuron. And then, of course, here is the axon of that presynaptic neuron. So some of these synaptic connections, you have this axodendritic connection, which is between axon terminals of one neuron and the dendrites of others. The axiosomatic is between axon terminals and one neuron and soma, the cell body, of others. Some less common connections, axon to axon, dendrite to dendrite, dendrite to soma. And the two main synapses are a chemical synapse and an electrical synapse. This is an animation that's found in the PowerPoint. Again, I can't play this one here. It does something weird in this recorded lecture. So do make sure to go in and review that animation about the neurotransmitters and how we can send that information along in the body. And basically, that's really important to sort of understand and view. And so neurotransmitters literally are the language of the nervous system. There are 50 or more neurotransmitters that have been identified, so there's a lot of them. Some of them we recognize more commonly than others, like acetylcholine and things like that. Most neurons make 2 or more neurotransmitters, so your neuron is making those neurotransmitters. And so neurons themselves, if they make more than, since they mostly make more than 1, 2 or more, they can exert several different types of influences. So it's usually released at different stimulation frequencies and usually classified by either their chemical structure themselves or how they function would be the classification of those. I've already mentioned acetylcholine several times. I mentioned it also back in the muscles. But acetylcholine is the first identified and best understood neurotransmitter that we have. It's released at the neurotransmitter junction, and it also may be used by ANS neurons and CNS neurons. And basically it's synthesized from acetic acid and choline by enzymes, the enzyme choline acetyltransferase. So it's degraded by the enzyme which we abbreviate ACHE, acetylcholinesterase. And so, again, this is the one that we have the most knowledge about, and it's the most understood. We also have the catecholamines. Dopamine is an example, norepinephrine, and epinephrine, which is made from the amino acid tyrosine. There's also the indolamines, things like serotonin, which is made from the amino acid tryptophan, and histamine, which is made from the amino acid histidine. All are widely used in the brain. They play roles in emotional behaviors and even the biological clock. So it's used by some ANS motor neurons, but imbalances of these particular types of biogenic amines, things like serotonin, histamine, catecholamines, dopamine, and all of epinephrine or epinephrine, typically is associated with a mental illness. What we're seeing, and especially understanding more, is that things like depression typically has to do with how serotonin is being taken up, and things like that. And so now we're understanding that the chemical structure and chemical nature, not only of the neurons, but how those neurotransmitters are sent via signals, either too much or too little, are affecting brain functioning. So it's very important. Amino acids basically make up all proteins. We've talked about that before. So it's really hard to determine which ones are actually directly neurotransmitters. So amino acids that are proven neurotransmitters: glutamate, aspartate, glycine, and GABA, which is gamma aminobutyric acid. And so those we know act as transmitters as well. So peptides also, those are strings of amino acids that have diverse functions. One called substance P is a mediator of pain signals. Endorphins, beta endorphin, dynorphin, and the encephalines act as natural opiates, so that reduces our pain perception. And of course, the gut-brain peptides, such as somatostatin and cholecystokinin, which plays a role in regulating digestion. Also, purines can act as neurotransmitters. So purines are monomers of nucleic acids, and they have an effect in both the central nervous system and the peripheral nervous system. So ATP, the energy molecule, is now considered a neurotransmitter because of how it acts. And ATP is a type of purine. Adenosine basically is a potent inhibitor in the brain. So caffeine blocks adenosine receptors. And it can induce ionic calcium influx in the astrocytes of the cells. Gases and lipids, so there's some gaseotransmitters as well. Nitric oxide, carbon monoxide, hydrogen sulfide gases. They bind with G protein coupled receptors in the brain. They are lipid soluble and are synthesized on demand. So NO involved in learning, information of new memories, as well as brain damage in stroke patients and smooth muscle relaxation in intestine. H2S, hydrogen sulfide gas, it acts directly on ion channels to alter different functions. The endocannabinoids, they act at same receptors as THC, which is the active ingredient in marijuana. And its most common G protein linked receptor in the brain. It is lipid soluble, and it is synthesized on demand. So it is believed to be involved in learning and memory, and may be involved in neuronal development, controlling appetite, and suppressing nausea. So neurotransmitters inhibit or exhibit a great variety of functions. So functions can be grouped on two basic classifications, whether they have an effect or their action. So the effects, is it excitatory or inhibitory? So neurotransmitter effects can be excitatory, which it refers to depolarizing, or inhibitory, which is hyperpolarizing. So the effect is determined by the receptor to which it binds. GABA and glycine are usually inhibitory. Glutamate is usually excitatory. Acetylcholine and norepinephrine tend to bind to at least two receptor types with opposite effects. So acetylcholine can be excitatory at the neuromuscular junction in skeletal muscle, but it can be inhibitory in cardiac muscle. And then, of course, we have that classification of action, direct versus indirect. So direct action, that neurotransmitter binds directly to and opens ion channels. So it's going to promote a rapid response by altering membrane potential. Examples are acetylcholines and amino acids. And then indirect action, where the neurotransmitter acts through intracellular second messengers, usually G protein pathways. So you have this broader, longer lasting effect similar to hormones. And these biogenic amines, neuropeptides, and dissolved gases tend to fall under this indirect action. Neuromodulator, that's a chemical messenger that is released by neurons that does not directly cause EPSPs or IPSPs, but instead affects the strength of synaptic transmission. So they may influence synthesis, release, degradation, or reuptake of neurotransmitter. They may alter sensitivity of the photosynaptic membrane to that neurotransmitter and may be released as a paracrine. So paracrine release is sort of the only room, if you remember, only FX is local. G protein linked receptors, their mechanism, that neurotransmitter binds to G protein linked receptor, activating G protein. And that activated G protein controls production of second messengers such as cyclic AMP, cyclic GMP, or even ionic calcium. So second messengers can then go in and open and close channels, activate kinase enzymes, phosphorylate channel proteins, activate genes, and induce protein synthesis. Neural integration, that's where you have neurons functioning together in groups. So groups can contribute to broader neural functions. And there are billions of neurons in the CNS, so must have some kind of integration so that the individual parts can fuse to make a smoothly operating whole. And then we have the idea of the neuronal pool, which there's functional groups of neurons. So they integrate incoming information received from receptors, kind of pool them together, and then forward process that information to other destinations. And then, of course, the developmental aspect. The nervous system does originate from the neural tube, and the neural crest is formed from the ectoderm. The neural tube becomes the central nervous system, so the neuroepithelial cells of the neural tube proliferate into nerve cells needed in embryological development. So things like neuroblasts become amniotic and migrate. Neuroblast sprouts exons to connect with targets to become neurons, and so on. Of course, growth is important as well as the developmental aspects. So there's a growth cone, and these growth cones are very prickly in structure at the tip of axon, so it helps it interact with its environment, helps it find those areas that it needs. The cell surface adhesion proteins, laminin, integrin, things like that will provide anchor points, and the neurotrophins will act or attract or repel that growth cone. And then there's a factor called growth factor NGF, which keeps that neuroblast alive so that development can happen. And so that's really important in embryological development and appropriate development of the nervous system and the fetus. So once an axon finds its target, it then must find a place to form a synapse. So about 2/3 of neurons actually die before birth. So that if the axons don't form a synapse with a target, they are triggered to undo or undergo apoptosis, which I think we've talked about before, which is programmed cell death. And so many other cells can undergo apoptosis during development. It's not just nerve cells. So that's just one type. During childhood and adolescence, of course, learning reinforces certain synapses and sort of prunes away others. So there's a lot of evidence that shows that genes that promote the synaptic pruning may predispose an individual to things like schizophrenia or something like that. So neurons are amniotic after birth. However, there are a few special neuronal populations that continue to divide. Those are things like olfactory neurons and the hippocampus. This is what one of those neural growth cones look like, and you see those little spiky things that help them attach. So that's just kind of a micrograph of what that can look like. So that's just a brief overview of that nervous tissue. And so make sure to go through all the information provided, look at the videos that are provided for you, and if you have any questions, please reach out because I'm always happy to help.